All right, welcome to our meeting. Oh, I just always call it a class meeting in my BYU one. Welcome to our 11 o'clock Bio 201 for Friday, April the 3rd, and hope that all of you are doing well and everything. So we're just gonna go ahead and get started into the nervous system. So I hope that you printed off chapter 11 notes on the fundamentals of the nervous system. So we're just gonna start out with some basics about the nervous system and then go from there. So let me find in my PowerPoint. All right, oops, I want to do that. Share. All right. Oh, and then I was going to mention, because we talked about this before I started recording the meeting, Annette has made some Quizlets of our information, so she's going to send me the link to that, and then I will post that link in the in an announcement so that you'll be able to benefit from her great hard work and her benevolence. So thanks for doing that. All right, so let's just go ahead and get started into the nervous system. So, okay. All right, so always refer back to the objectives when you're preparing and studying and reviewing and everything, because this is what I want you to know. So first thing that we're going to take a look at are the basic functions of the nervous system. So a lot of times we think the nervous system is super complicated and hard to understand, and it is kind of weird to be thinking about how you think. You know, that's like contemplating your own existence and everything, which is a little existential, but anyway, there are only 3 functions in the nervous system. So everything that we do, we can put into these 3 categories. Is it sensory input? Is it integration? Is it motor output? So what we have with sensory input is that we have something that is not in homeostasis anymore. So when we see, we would think, well, what if nothing's moving? Well, no, it's still not in homeostasis, because we have to keep, our brain doesn't keep the images unless we put it in our memory, but our brain doesn't keep the images and just gets rid of them. So when we're seeing things, it's a lot like, you know how an animated picture is. You draw each frame and then you speed it up and then make it look like things are moving. That's kind of how our brain sees things. And so even with vision, our eye position is changing, whatever, and so everything is out of homeostasis. And so when things are out of homeostasis, then we need to perceive that they are, or we need to receive the fact that they have changed. And so this person looking at this glass of water sees the glass of water. Maybe it might have an odor to it. Maybe it's not water. Maybe it's some other kind of beverage that has an odor to it or an aroma to it. And so that would also be detectable. And so then we're going to collect that information. We're going to send it to our brain and spinal cord. So as we collect that information, this input to the central nervous system is called sensory input. And sensory input only travels one direction, from out in the periphery to the central nervous system. This neuron right here, this blue neuron, blue for now and ever in this course, well, even next semester too, blue is always sensory. So sensory neurons, we always draw them blue just to keep track of them. So sensory input always uses sensory neurons that are going to, like I said, collect the information and move toward the integration center, which is the brain and spinal cord. So this is where information comes in, and then this is where we process the information. So integration is thinking. It's deciding what we're going to do. Now, some of our thinking is very, very simple, where you just have one neuron coming in, one neuron deciding what to do, and one neuron sending that in, and that same neuron sending the information out for your response. And that would be like a reflex arc. Very, very simple. But in this integration center of the brain and spinal cord, or the central nervous system, that's going to be an interneuron right here. Interneurons are always green, and they're going to be collecting sensory input and shuttling it off to somewhere else. They may send it to another interneuron to, you know, get help with, well, what are we going to do about that? Well, I don't know. Let's think about it. Let's go ask Joe. And then they send it to another interneuron, and so we can process information in that manner. But then when we're ready to respond to put us back into homeostasis, then it will be motor output that does that. And we've already discussed motor output when we were talking about motor neurons with motor units. So all motor output can be, oh, you know what? I'm going to write that down. So, you know what? I'm going to type some stuff in here. I just thought, because I usually write on the board for this, but okay. So this is going to be, oops, sensory neurons from receptors. Oops, from receptors. Okay, so that's our sensory input. Sensory neurons from our receptors. Then we're going to have integration. So this is processing. So let's do it this way. Interneurons process incoming information, and then they I guess we'll put stimulate effectors. Okay, so the effectors are the things that are going to do the response. And so then the motor output, so motor neurons, oops, carry, oh, let me put some more information in all of this. Okay, so motor neurons carry from CNS to effectors, and then the effectors are muscles and glands. And that's kind of crazy to think about. The only responses that we ever do are with the three kinds of muscles that we have, so cardiac, skeletal, smooth, and the glands of our body. Every single response. There is not a response that doesn't go to one of those places. So the motor neurons that we were talking about with motor units, going to skeletal muscle, there's also motor neurons that are involved in influencing how the heart contracts, not telling the heart to contract, but influencing its action. And then we also have autonomic nerves that tell the smooth muscle to contract, that act at the level of the smooth muscle to wherever it might happen to be to get it to do its job. So yeah, all of our motor output will always be going to a muscle or a gland. The other thing that I wanted to put up here is that these may be called afferent neurons, and these may be called efferent neurons, because afferent means to carry toward and efferent means to carry away. Okay. All right. Oh, let's go ahead. There is another name. I like almost never use it, but oops. We can also call these association neurons. All right. So I think that's oh, let's do one more thing. Sorry. So where does this take place? Where does integration take place? In the CNA. In the CNS. And then this is going to take place in the PNS. And then this is also going to take place in the PNS. Okay, so there is everything. And if you can get this little pathway down, it's going to really help your understanding of how things work when we get a little more complicated than something just as simple as this. Okay, so I hate to clear all of that, but I can't go on unless I do. So there we go. All right. Now, some of the next information that we're going to do, where's my thing? There it is. we're going to do in lab, but actually I do want to look at this first. So this has the whole organization of the nervous system. So we just saw that we have the afferent or sensory division, and this is going to be in the PNS. So all of this in yellow is PNS, and then the CNS is the gray, central nervous system. So PNS stands for peripheral nervous system. So we've got nerve fibers, which are just axons of neurons, okay, just like we have muscle fibers, so neurons can also be called nerve fibers. And so what do we have? We have somatic, that's going to come from our body, and like our skin and our muscles and the outer parts of our body, and then visceral would come from our internal organs, like our, our heart and our lungs and our liver and kidneys and stuff like that. And then their job is to conduct impulses from the receptors to the CNS. Okay, then we get, like we saw, we get to the CNS, and that's going to be made of the brain and spinal cord. So you can see orange is the anatomy, and green is the physiology, is the function. Okay, so what is the function? Integrative and control center. So this is where all the input comes in so that we can decide what output goes out. Oh, I skipped peripheral nervous system. So all the cranial nerves and the spinal nerves, everything outside of the brain and spinal cord is part of the PNS, peripheral nervous system. And that just has axons. There's some dendrites out there, some cell bodies, but it uses the cranial nerves and spinal nerves. And their job is communication lines between the CNS and the rest of the body. So this input goes through the PNS, sensory input goes into the PNS, into the CNS. Then the motor output goes through the PNS and then out to the motor division. So these will be motor neurons. And like we just said, they conduct impulses from the central nervous system to the effectors, which are muscles and glands. We're not going to worry too much about this because we have an entire chapter on the ANS and sympathetic and parasympathetic divisions. Somatic, though, is our body. So this is going to be all of our voluntary muscle. So our somatic nervous system will carry those impulses from the conscious part of our brain, from the cerebrum, down through the PNS to the skeletal muscle. So all the motor neurons that you know of so far have all gone to the PNS and, gone to skeletal muscle. So those have all been voluntary. So it's all been part of the somatic nervous system. Okay, now, okay, so here is just a nice short little summary of the functions of the nervous system. Monitor changes inside and outside the body, which are stimuli. Gather the information as sensory input, and then integration, processes, interprets what should be done, and then causes responses by activating effector organs, motor output. Okay. All right. Now, this stuff on supporting cells, we're going to do in lab on Tuesday. So that sounds kind of funny, huh? We'll do this Tuesday afternoon in our time designated for lab. So we'll do that, that. This stuff, all this structural, because this is all anatomical. So we're going to do all of this and this. All right, so we're going to go right to neurophysiology. And guess what, friends? Most of this is identical to what we have learned already in anatomy, in the muscular system, which makes it really super nice. You know, it's nice that things have an economy of function. I like that once you learn basically how things function, it applies in a lot of situations. So you just have to learn it once, and then you just look at how it's modified to fit specific situations. But you can get a handle on how it works in general, then you'll be fabulous on how it works in specifically. So we're going clear back to membrane potentials that we learned already and apply the membrane potentials now to neurons. So we have membrane ion channels, just like exactly the same ones that we had when we were talking about membrane potentials, and exactly the same ones that we had in the sarcolemma of the muscle cell. So We have two kinds. We have active gates. We just kind of changed the terminology a little bit. So active gates are ones that open and close. And we already know that we had the chemically gated or ligand gated channels, which have the receptors for the neurotransmitters, and they'll open when the neurotransmitter binds. Then we have the voltage gated channels that will open in response to the change in the membrane potential. So when it gets to a threshold, when it gets to -55, then it's going to open up. Same as we've always learned. Okay, then we also have passive gates. Those are the ones that are the leakage channels, the ones that are always open, and always we have a channel specific for each particular ion. So we have sodium channels, we have potassium channels, we have chloride channels, we have calcium channels. So whatever we need to, we even have water channels, which we'll talk about next semester. Whatever we need to move across the cell membrane, because really the cell membrane provides an impermeable wall. If it was just phospholipids and no proteins in the phospholipid bilayer, very few things would get through. Respiratory gases and a little bit of water and anything that's lipid soluble, but everything else that we need would never, ever cross the cell membrane. So it's really a good thing that we have proteins in the cell membrane. All right, so there it is. You guys know this. Here's the chemically gated one. So as soon as the receptor binds, there's the acetylcholine, then the sodium comes in. Remember, we do have a little bit of potassium that goes back out, but we're not going to stress about that. We're just going to focus on the sodium that comes in, because we need that sodium to depolarize the membrane so that we can create an action potential. And then we create an action potential with the voltage gated channel. So as soon as we look at on the left picture, it's plus on the outside, minus on the inside. As soon as we reverse that, so we depolarize the cell membrane, then that's going to open up the channel. There, the channel's open. Here it's closed, and the sodium comes in. Nothing different from what you already know. Resting membrane potential, exactly the same as what we had in before. -70 millivolts. What establishes it? The leakage channels, and what maintains it is the sodium potassium pump. So here, just, you know, there's the -70. This is everything you've seen before. But what are the ions then that are involved? So lots of sodium, lots of chloride on the outside, lots of potassium, and the anions. Look at how few anions. Here we have 100 millimolar anion concentration. Here we have a 0.2. So this is 2,000 times smaller than what we have out here. Crazy, 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 okay? Not 2,000, 500. I went the wrong, I multiplied it instead of dividing it. Or is that 5,000? No, that's 5,000, right? Times 10, 5 or 500. No, 5,000 would have to take it to 100. So crazy. Don't worry about my math. Don't worry about your own math. Okay, and then, so lots of potassium. I'm an idiot. I wish I could edit that out, but I don't know how. So potassium and sodium. No, potassium and the anion. Okay, I'm just going to quit while I'm ahead. Here's the sodium potassium pump. We know that potassium's diffusion is to the outside and sodium's diffusion is to the inside, but without channels, that can't happen. And so let's say we put some leakage channels in here, and that's what's going on. So now we have to restore it. Now we've got to pump the sodium back out. We have to pump the potassium back in. All right. Same as always, depolarization is a reduction in membrane potential, but it gets less. There's less of a difference between the outside and the inside. So that -70 number gets not as negative, -55, -40, 30, whatever. And as we depolarize, it increases the probability of producing a nerve impulse. Now, there's going to be something slightly different about neurons than what we see in a muscle cell. So with a muscle cell, the motor neuron is going to stimulate the muscle cell. If enough acetylcholine comes in to stimulate the muscle cell, then the chemically gated sodium channels open up. I mean, they do, but enough sodium would come in to take the membrane to -55 and it would depolarize. If I don't have enough acetylcholine crossing the gap, then of course it's not going to depolarize. In a neuron, it's slightly different. The way a neuron is designed, it has the ability to collect impulses from several different places. If you notice, one end of a neuron has lots of branches off of the end called dendrites, and the job of those dendrites is to collect sensory input. Now, a lot of the sensory input that comes into our nervous system is subthreshold. If just that one axon was carrying the information to the neuron, it probably wouldn't fire. It would probably just, you know, be subthreshold. But if you had a bunch of subthreshold stimuli coming in, so a bunch of axons, neurons from little places coming around and acting on the neuron, then finally the neuron would fire. Okay, it's kind of like when that one little you've probably seen that Family Guy video where Stewie's trying to wake Lois up and he's going, Mom, 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 Mama, Mama, Lois, Lois. Anyway, YouTube it if you haven't seen it. And so he's just, you know, standing by the side of her bed and he keeps calling her and calling her and calling her and trying to wake her up. And then all of a sudden she goes, what? And he goes, hi. And, you know, he didn't want her for anything. He's just being Stewie. But that's how your neurons work. A lot of subthreshold stimuli until finally they all summate. Hey, we know what summation is, to produce a nerve impulse. Okay, so we're going to come back to that and discuss that a whole lot more in the future. All right, this, you know what this is. Depolarization, less negative, hyperpolarization, more negative. So hyperpolarization occurs when the membrane potential increases. It gets more negative on the inside than at rest. And hyperpolarization reduces the probability of producing a nerve impulse. Are there times when we don't want our neurons to fire? Well, yeah, I don't want my neurons to fire when I'm asleep. I don't want to be aware of my surroundings. I want those neurons to fire that keep me alive while I'm asleep. But the neurons that make me aware of my surroundings, I want those hyperpolarized. And so whenever we are not aware of something, it's because the neurons that would be collecting that information have become hyperpolarized, or they are hyperpolarizing at the integration center. So they get up to your brain and then you get hyperpolarized up there in your brain. Really good example of this is in your arm. I mean, just super, super simple. So when we contract, or when we flex our elbow, we want to contract the biceps brachii. So we're sending a depolarizing stimulus in the form of acetylcholine to the muscle fibers in, or to the motor neurons actually, that innervate the biceps brachii muscle. But at the same time, my spinal cord doesn't want my triceps brachii to be contracting. And so the motor neurons that go to my triceps brachii are being inhibited or hyperpolarized by a neurotransmitter called GABA. So at my spinal cord, my interneurons, remember brain and spinal cord, integration center, to those neurons that I want to fire, I'll be sending acetylcholine for stimulatory impulse, or whatever, stimulation. But then I'll be sending GABA with my, from my interneurons to inhibit and hyperpolarize my, my biceps, or my triceps brachii. And again, we'll talk more about that, but that just gives you how it works. Okay, so we do want to, some cases we want to stimulate something, but we want to inhibit also. All right, and that's hyperpolarization. Okay, so remember I told you that we can have a bunch of subthreshold stimuli, and in neurons, those subthreshold stimuli can all add up together and end up producing a nerve impulse. So we call those little baby depolarizations graded potentials. So graded means they come in different levels, different amounts, different strengths. So they'll depolarize to different levels. So let's say we know that if we want a threshold stimulus to happen, then we need to have a stimulus of plus 15. So it has to take minus 70 to minus 55. So that would be plus 15 millivolts worth of sodium coming in. Well, sometimes I can't get that much coming in. So I might have enough sodium that would take the membrane to plus 3 in one place, and then would take the neuron to plus 5 in another place, so that's plus 8, and then a plus 4 somewhere else, so that'd be plus 12, and then a plus 3 potential, and then that would be 15. And so all of those depolarizations can then add up and then cause a nerve impulse to occur. And then we can get the same thing with a hyperpolarization. So maybe we have a stimulus that causes more potassium to come out, and in one place my membrane drops to -6, or was 6 lower, so now it's -76. So a +15 stimulus is not going to open up my membrane because I'm still going to be 5 short, 6 short. Okay. Don't worry, you can watch this again, but I will go over this again lots and lots and lots. Okay, this is going to be the exact same thing as what we saw in a muscle cell as far as how does depolarization go along a membrane. So no matter where I depolarize it, then I've got voltage gated sodium channels all entirely around that area of depolarization, so they'll all be depolarized. And so I'm creating an action potential here as my depolarization spreads over my membrane. Here, if I depolarize right here in the center, you can see that depolarization is not going to keep going if I don't have voltage gated sodium channels to boost the signal. So it's kind of like having, let me draw in here, hang on. Okay, so if I don't have voltage gated channels along here to boost the signal, like cell phone towers, the signal's gonna peter out. Okay, so and sometimes that happens. So in dendrites, for example, dendrites don't have they don't have voltage gated sodium channels in their membranes. They just have chemically gated ones. So let's say we want to get to -55 right here. Okay, and then the axon's over here. Okay, so an axon over here, because we're just going out from one spot. So we're just running down the cell body to an axon. All right, so if I depolarize it right here, yeah, that might be slightly over -55, but look at what happens as I work my way down to the axon. My signal peters out. So if I want this little guy to be the only depolarizing signal that I have in my axon, I gotta go way up here. So if I follow the same slope, oops, so that I'm at -55, that initial signal has to be a whole lot higher. Okay, so that's why it's important that we be able to collect the signal so we can stimulate the neuron in a bunch of different places and have it then take the value of that depolarization to -55. And again, we'll talk more about this next time we're here, just so you're not confused. All right. All right. Oh, so here we are, action potentials. So the events are identical to those in a muscle cell. We also called it now a nerve impulse. So anytime that I use the phrase nerve impulse, I always mean an action potential down the axon, okay, down the long skinny part of a neuron. Only axons can generate a nerve impulse because this is the only place that has voltage gated sodium channels. So let me just make a note of that. Okay, so so voltage gated channels, we'll just put it like that, only found here, only found on axon. Okay, so we don't find voltage gated sodium channels and potassium channels anywhere else but on the axon. So channels along the axon open in response to changes in potential that occur along the dendrite and cell body membranes. This transition from a local graded potential to an action potential occurs at the axon hillock in motor neurons. Okay, so remember that, and let me just show you where the axon hillock is. So, all right, So let's go back to the structure. Oh, we can just look at this one. All right, so in this particular picture here, we're going to have a signal come and stimulate that neuron or that dendrite and that dendrite. And maybe I'm going to have one that stimulates my cell body. And so I get all these little baby depolarizations. So this is maybe a +3. Oh, that's an ugly looking 3. And a +4, +5, +2 in this case. Okay, right here is the axon hillock. Okay, so I'll go ahead and type that in. Okay. All right, now down here through the rest of the neuron, we're going to have voltage gated sodium channels all the way down the axon. But they don't start until right here. And the depolarizing stimulus, oh, gross looking. Okay, the depolarizing stimulus must get to +15, okay, to get this area of the membrane to be -55. So 7, 12, 14. Okay, so my total stimulus would be +14, and no, those voltage gated sodium channels will not open up, and I will not have an impulse. But if I took this one to 3, this would then go to 15, and then I would get an action potential or a nerve impulse traveling down the axon. All right. So that's that right there. Okay, and then this just, you can take a look at this. This is just in the resting state, depolarized, repolarized, hyperpolarized. And then we've seen this graph before. When we've seen this graph before, it didn't show the relative permeability, that P to sodium, so permeability to sodium, permeability to potassium. It didn't have those labeled. Blue is our action potential. Okay, so we're used to the action potential graph that we've drawn 100 times. But then we want to look at what's the permeability during each part of the graph. So we know during this depolarization event that we're very permeable to sodium, and then all of a sudden we stop being permeable to sodium and it just plummets back down. Whereas with potassium, we know that the potassium channels are very slow to open, and I would probably peak that a little bit over here, so it comes more in line with when the potassium gates start to open up. But you know, as more gates open up, my permeability increases, so maybe that's a good thing right there. And then we're right here and we can see, oh yeah, I'm still permeable to potassium. That's why I get this hyperpolarization right here. And then look at what happens right here. My cells permeability potassium drops way down, and then that's when I return back to resting membrane potential. Okay. And then this just shows what's happening to the gates as you go down. So that's kind of a cool little thing, but anyway. All right, now this is super important, all or none phenomenon. Okay, once you depolarize a membrane, it will completely depolarize all the way to the end. You'll get the action potential all the way to the end, and that +30 value will always be the same. You'll never get any more than that, and you'll never get it happening any faster than that. That it's just, once it opens up, then there it is, no matter what the strength was to begin with, okay? So, and I've shown you already, not all local depolarizations produce action potential. So not all those stimuli that come in on a dendrite are going to depolarize the entire neuron. They'll just depolarize that one spot, but it won't end up going all the way down the axon. So we said those depolarizations must reach threshold between +15 and +20, you know, +15, we know that. Subthreshold stimuli, so those are those brief weak stimuli. So just those little subthreshold stimuli are not translated to a nerve impulse. Stronger stimuli depolarize to threshold. Quick, weak ones must be applied longer so they can summate. But once threshold is reached, an action potential occurs along the entire axon length with the same amount of speed. It will not arrive at the end of the axon terminal, any faster in a strong stimulus than it is just barely threshold stimulus. Doesn't make any difference. Okay. All right, we have refractory periods, just like in a neuron, just like in a muscle cell. So When depolarized, the neuron cannot respond to another stimulus. So the absolute refractory period is the period from the opening of the sodium channels to the resetting of the gates. Each action potential is a separate all or none event, and the action potential is in one direction from the beginning of the axon to the end. Oh yeah, here's a difference. Remember, in a muscle cell, if you stimulated the muscle in the middle of the membrane, then it would go both directions all the way down the muscle cell. Doesn't do that in a neuron. It's always got to start at the receptive end where the dendrites are. It goes cell body axon. There are no chemically gated sodium channels on an axon, so you can't synapse with it to make it fire, and it's not going to go backwards. So you stimulate an axon hillock, it'll go down. You stimulate it anywhere else along the axon, nothing's going to happen. Okay, so it's got to go dendrite, cell body, axon. And then there's just absolute refractory period, relative refractory period, like we already know. And then the relative refractory period will be when the sodium gates are closed, potassium gates are open, and hyperpolarization is occurring. We already know a threshold stimulus at this point won't cause an action potential, but an exceptionally strong one can reopen the sodium gates. Okay, because we're at -80 maybe, so now I need a threshold stimulus of +25 instead of +15 to get it back open. And there's that again, just like we know. Okay, I'm trying to decide. Let's, yeah, we already know this, so let's just go over it. We probably won't go the whole 50 minutes today because I'm just about done. Sweet, it's Friday. All right, so the synapse. There are some terms that I want to make sure, because I'm going to use them over and over again. So make sure you know presynaptic neuron and postsynaptic neuron. So let me draw you a presynaptic neuron. Here's my presynaptic neuron. It's going to have my little synaptic vesicles. This would be my motor neuron in a muscle, you know, at the neuromuscular junction. And then here would be an axon, nope, a dendrite. So this is my presynaptic neuron. So it's before the synapse. This would be my postsynaptic neuron. Okay, and so it would have chemically gated sodium channels, oops, all in it right here. And then the action potential would travel this direction, and then the neurotransmitter would cross the gap. So we went from an electrical stimulus to a chemical stimulus, and then another electrical stimulus as the action potential travels down, or the depolarization travels down the or down the dendrite. Okay, so that's, make sure you know the difference between presynaptic and postsynaptic neurons. All right, here are the 3 places where a synapse can happen. This is the usual one right here, is the axodendritic synapse, when the axon of one neuron synapses with the dendrites of another. Or you can have the axon synapse right here with the cell body, because there's going to be chemically gated sodium channels here. And you can have an axoaxonic, where we're right here at the hillock, where we're just transitioning from chemically gated sodium channels to voltage gated ones. But remember, if an axon is stimulating it, then it's going to be a neurotransmitter that's crossing the gap. So there's got to be some chemically gated sodium channels there in order to for the neurotransmitter to bind to it. And then this just shows what a synapse looks like, a little chicken foot thing right here. So the axon's got all these little terminals down here to increase the surface area for reception. And then this is the cell body of a neuron. Okay, so transfer across the synapse can be electrical through gap junctions with connexin proteins, or chemical as discussed here. So let me show you just really quickly what an electrical synapse looks like. We'll never really talk about them anymore. We will talk about kind of their, you know, how they work with heart muscle cells, how they kind of act like electrical synapses. But here's going to be one, so this is the axon of one cell. And then here is the axon of another cell. So this is the pre. Ooh, well, that's the R now. Pre. This is the post. Okay, so what I'm going to do is, put some little channels in here, little gap junctions. So the cells are actually touching one another. And then they have gap junctions. Oops. Oh, that'll work. And then we'll put sodium in here. And then the sodium will pass through here. So instead of sodium coming in from the outside, it's coming in from the presynaptic neuron. And so those are electrical synapses. Okay. Otherwise, we've got the good old chemical synapses like we know. So let's look at the picture and we'll go back to the description and then we'll be done for today. I think that'll be good. All right, so a nerve impulse comes down the axon. So this would be a motor neuron coming from my brain. Let's write that in there. Okay, so this is a motor. Well, it doesn't have to be. No, I don't want it to be a motor neuron. It's just a neuron. Okay, so nerve impulses coming down from the neuron. Could be a sensory. This is actually, let's make it a sensory neuron. So this is a sensory neuron carrying the information from your eyeball, for example. Okay, so as that nerve impulse gets down here to the bottom, it's going to open up the calcium, the voltage-gated calcium channels, and calcium will flood into the cell, triggering exocytosis. Okay, so the synaptic vesicles, Okay, so these little synaptic vesicles will come and fuse in step number 2. The acetylcholine will come out. It'll bind to the receptors. Look at, see, these babies are closed because the neurotransmitter hasn't got to them yet. This one's closed because the neurotransmitter is not there. Same with that one. But all the ones that have neurotransmitter in them are all open. So here the neurotransmitter binds to the receptor and the sodium rushes in. And then once the acetylcholine is gone, then the sodium can't come in because the ion channel closes. So let's go back to those notes. So calcium gates open when the impulse reaches the axon terminal. Voltage gated calcium channels open and calcium acts as the messenger to cause the synaptic vesicles to bind in exocytosis with the presynaptic membrane. Calcium pumps are going to be there too. So as soon as the calcium comes in, we're going to pump the calcium back out. Okay. Then the neurotransmitter binds. After diffusing across the synaptic cleft, it binds reversibly to the receptors clustered in the postsynaptic membrane. So it doesn't stay there forever. It leaves. It's broken down. Something happens to it. We'll talk about that on Monday. And then the last thing we'll do right here is a local potential is generated as the ion channels open up. This may excite or depolarize or stimulate the neuron or inhibit or hyperpolarize the postsynaptic neuron. The more intense the stimulus, the more neurotransmitter is released, and so more likely will these open up. But once they open up and get to threshold, boom, then the neuron will fire. So if we wanted to draw in, okay, so right here, this is going to be plus, 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 and then minus, 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 minus. But then right here, now it's minus, 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 and plus, 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 because we just created that local potential. Okay, I think, yes, that's where I want to stop because I want to talk for a little while about termination of neurotransmitter effects. Oh, and then I have to show you a synapse thing. I don't want to forget about that. Okay, so that will do it for today for our, hang on, Oh, there we go. Like, how do I get out of here? Okay, so that does it for our lecture today. Make sure that, well, I don't know. Let's see, you finished the quiz. Oh, let's just go find out because I've got so many different things going on. Let me find out when you need to make sure you have your quiz done by.
Mrs. G?
Yes.
I hate to be that guy, but last time you talked about us going over the structural characteristics
Of
and we didn't go over them.
structural characteristics of what?
We were talking about metabolic characteristics and structural characteristics.
Oh, I didn't do that.
Muscle, yeah.
Oh, yeah. Okay, you are that guy. Thanks. Let's do that right now because it's not long. Okay. Thank you, Annette. Okay, let me find that document. Yeah, don't worry, because people can leave. It's not like, you know, they gotta stick around and you're not a bad guy. All right. Let's see, where's that? Okay. Muscle fiber types. All right. Okay, and we're gonna go here to share my screen. All right, yes. So let's go ahead and finish this up. Okay, so in our slow oxidative fibers that we've been talking about here with their metabolic characteristics, now we want to take a look at, well, what would we see? What would they look like if we looked at them under the microscope? Okay, first of all, and we don't even have to look at them under the microscope really to see mostly this. So what is their color? Well, if you look at a steak or a roast or, you know, whatever, it's pretty red as far as beef is concerned. Even in a chicken, it's kind of, you know, you have dark meat and light meat. Okay, so the red is the dark meat in a chicken. It just isn't as red as it is in a cow. All right, so why is it red? Well, if we look up here, let me find my little drawing pen things. Okay, so if we look up here, oh, what do I have a lot of? Have a lot of myoglobin. Okay, now remember myoglobin holds on to oxygen just like hemoglobin does, and when hemoglobin hangs on to it, it's red. Okay, so that's why slow oxidative fibers are red, is because they contain a whole lot of myoglobin in them. All right, what about their fiber diameter? Well, they're very small in diameter. In fact, I'm going to make a little comparison. So they're going to be like this. Now, what does that mean? Not many myofibrils are inside. I'm not looking to produce a lot of force. I just, you know, need it to contract. Okay, so I don't need a lot of strength out of these. And so my small muscle fibers, so I'm going to put slow oxidative. Okay, so those guys don't have a lot of myofibrils inside of them, but they have tons of mitochondria. We would be able to see that. Why do they need tons of mitochondria? Because what are they doing? Aerobic respiration. Okay, so they are carrying out cellular respiration. And where does that happen? In the mitochondria. Now, what do they need? They need O2 and food. So what's going to bring them the O2 and food? Capillaries. So we have lots and lots and lots of capillaries that are running through slow oxidative muscles. Okay, muscles that have primarily slow oxidative fibers. All right, then if we go to the fast oxidative fibers, what color are they? Well, they're red to pink because they have a little bit less, okay, so just slightly less myoglobin in them than what the slow oxidative fibers have. Okay, so they're going to be red to maybe a pink color. Okay, not quite as red as the other ones. What about their muscle, their fiber diameter? Well, they're going to be a little bit bigger around. Okay, they're going to contain slightly more myofibrils because, so these are the fast oxidative, because we need a little bit more force out of them, okay, than just, you know, slowly walking, whatever. Okay, so I need a little bit more strength from those. So they're going to have an intermediate fiber diameter. They're still going to have many mitochondria and many capillaries because they still can carry out aerobic respiration. But then we have the, I should have done them, and I'll do them in orange. Okay, then we have the fast glycolytic fibers. Okay, fast glycolytic fibers are white. Now, we don't really see that in a cow. We don't see a separation between muscles that are primarily fast glycolytic versus muscles that are primarily slow oxidative. They're all pretty mixed in, and the red just kind of overshadows the rest of the muscle fiber types. Okay, but we do see it in poultry. Okay, we see it in chickens and we see it in turkeys. And the reason why is because we have bred those birds so that they have muscles. So they're muscles that normally would have a lot of fast glycolytic fibers. They're really powerful muscles. Okay, we have bred them so that they are only fast glycolytic fibers. So if you think about where the white meat is in a chicken, okay, we needed some fast glycolytic so that we could fly, we could get a big burst of speed, but then we also would need some slow oxidative so we could keep that speed going. But you know what? There were some birds that had more glycolytic fibers, and somebody went, wow, I like the taste of that. That's a little bit different than what we've been eating in these game birds like pheasants and wild turkeys and ducks and those kinds of things. And so they started breeding these chickens together with white meat, you know, with more white fast glycolytic fibers, until all of their breast meat and their wings were entirely fast glycolytic. So, you know, the chicken can flap its wings pretty hard, but it can't go anywhere because it can't produce a lot of force, because we took most of that away. So we made it so they can't fly for, you know, over distances or whatever. But we did that because we like the way it tastes. But we don't let our chickens lift weights, so it doesn't get really big like it can in humans. But remember what I said last time when I said these are like in your pectoralis major muscles. So this is fast glycolytic. They are big around because they have lots of myofibrils inside. So lots and lots and lots and lots and lots of myofibrils. And remember when we weight lift that we make more myofibrils? So these are the cells that hypertrophy. So whenever we see big muscly people, they have been selectively targeting the fast glycolytic fibers within those muscles to build those up, because they're the ones that are going to hypertrophy. These guys are not. I don't get hypertrophy in slow oxidative fibers. They don't build bigger than what they already come. So the cells are big around. They can produce a whole lot of force if you strengthen them. And so they have few mitochondria because what are they doing? Anaerobic respiration. They do not need oxygen. And so they have few capillaries too, because they store their glycogen within their cells. Okay, so you know, at Thanksgiving when I teach this, I usually say, okay, who likes red meat? Who likes dark meat? Or light meat, you know, white meat and dark meat. The dark meat, where do we find it? We find it in the legs, and the thighs of the chicken, because why? They need to run away. And they're not flying, but then we bred out their ability to fly. We just left them with these white ones with nobody else to help them out. And because you don't have a weightlifting chicken, they can't, you know, be like The Rock or somebody like that. So if you don't like the taste of red meat, it's probably because you don't like the taste of myoglobin. If you don't like white meat, it's probably because it's too dry for you, because there's not enough like blood vessels and stuff in it. So I'm a dark meat person. I don't really like white meat at all. So anyway, unless it's like rotisserie chicken or super basted or something like that. Otherwise, I won't eat white meat. Ugh. Okay. All right. So that finishes up that chart for you. So now you've got everything that you need, and we should be good at this point. So I'll stop sharing that. And if you have any last minute questions before we go, I have a test at 1 o'clock I got to go take. So I'm going to bust out of here because I got to go home and take it on my home computer. Otherwise, I see people leaving, so nobody's got a comment. So that's great. So I'm just going to go ahead and stop the recording, and then I will see you on Monday. Bye. Have a good weekend.